previous. Okay, so I am a scientist, not a clinician, but I've been working for about the last 15 years on my baby, which is really developing blood tests for breast cancer. There are currently no blood tests for monitoring breast cancer, so once you've been diagnosed and treated, it's a bit of a black box as to whether your treatment will be successful or not. That's really analogous to treating diabetes without monitoring blood sugar levels. So diabetes is successfully managed because we can monitor the sugar levels. So why not do the same thing with breast cancer, monitor patients' response to treatment, and then hopefully we will improve outcomes in that way. So I've been working towards new blood tests for breast cancer for the last 15 years, and a lot of my research has been in collaboration with Imperial College and with clinicians based there. And I've been extremely fortunate to get substantive funding from Cancer Research UK, from their Experimental Cancer Medicine Networks, or ECMC, and Lynn at the back's representing that network today, and then locally from Hope Against Cancer with Julian Barwell, where we're focusing on your group, the cancer families. So just a few simple slides. I've tried to make this very simple and basic and not overwhelm you with high-powered science. So this is a cartoon which helps me to tell you that cancer DNA is found in the blood. On the left-hand side here, I've got a representation of a cancer or tumour. And these sort of circles, these blobs, represent individual cells in the cancer. Now I've got purple ones and blue ones, because a cancer isn't just a homogeneous mass, it's a combination of different cells and cell types. And some of them are naughty, we don't like them, they're what causes problems. These are the purple cancer cells. Some of them are blue, these are normal cells as well. So there's a mix of normal and cancer, even within a tumour. And amongst these cells, occasionally they die from natural causes. And when they die, they shed their DNA, these kind of little coils here. That DNA is shed and that can get into the blood. So on the right-hand side, here's a cartoon representation of a blood vessel. And within that blood, we've got the normal blood makeup, we've got our white blood cells which defend our bodies against attack. We've got these nice little red blood cells which carry oxygen around the body, and all this sits in a nice soup which we call plasma or serum. And within that, occasionally, we will get other cells coming into the blood, both our normal cells and then our cancer cells as well. So we've got the cells occasionally occurring in the blood, and we've also got this shed DNA occurring occasionally in the blood. Now I've coloured these cells differently, because they are different. Not only are these cancer and these healthy, but their genetic makeup is quite different. And as Joyce presented earlier, we now know there are 10 key genetic makeups for breast cancer. I'm sure actually the picture's much more complicated than that, and the 10 probably branches out into many hundreds of individual differences. But there are 10 main subtypes. And the DNA is sufficiently different that we can tell cancer from healthy. So my research has really been focused on detecting this cancer DNA, which is shed into the blood if you have cancer in your body. So the analogy I like to use is with forensic science. Forensic science in recent years is predominantly based about identifying individuals from minute traces of material. It's based on something called Lockhart's principle. Every contact leaves a trace. So when you touch something, you may leave behind 1 to 2 cells. A forensic team can come in, swab that from those 1 to 2 cells, get out the DNA. From that DNA, identify the individual. So by analogy to this, if you've got cancer in your body, 1 to 2 cells might release that DNA into your blood. If we know what to look for, we can detect it and we can characterise it. And we've been successful in showing that we can tell cancer from healthy by blood tests. And we published this work last year in a journal called Human Research. And this is just one scientific piece of data. That's all I'm going to show you in terms of real science, really to illustrate this fact. So we took the DNA from the blood 65 women with breast cancer, and then we took DNA from healthy women, women with no evidence of cancer. And we profiled this using highly sophisticated techniques where you can look at all of that DNA in a single experiment. We looked at 1.8 million data points in each sample, and we analysed over 300 samples in total. That took us 2 years to analyse that properly. But when we did that, we found that cancer and healthy split. So we can tell cancer from healthy by looking at the blood. And if you look on the left-hand side, we've got these sort of jaggedy lines. This is the DNA profile in a patient with breast cancer. Along the bottom, this represents human chromosome 10. So this is all of the long arm of chromosome 10. We've got 4 different DNA samples from this lady. This top profile is her normal DNA taken from white blood cells. So these are healthy cells, no evidence of cancer. You can see this sort of jaggedy line representing normal variation along the chromosome. We see local copy changes. So some areas of the chromosome show a little bit of gain, some areas of the chromosome show a little bit of loss. That's natural human variation in everybody's DNA, and it's a characteristic of everybody in this room. But then when we looked in her cancer tissue, so her tumour at the bottom, we've got this massive peak or spike here, and then another one over here on the left. And these peaks or spikes are completely absent from her normal DNA. So these are cancer-specific local changes to the DNA. And when we looked in the blood, we've looked at 2 different blood samples. This sample again has this cancer-specific spike. The second cancer again has the cancer-specific spike, and also evidence of new spikes developing as well. We believe that this is evidence that this woman had persistence of disease and potentially she was developing metastases because her cancer DNA was changing. So we potentially have tests amenable for real-time monitoring of cancer through analysis of this DNA. Not every patient has these profiles. On the right-hand side, this is another lady. Again, we've got her normal white blood cell profile across the top, typical of normal healthy DNA profiles. Again, in her cancer, here you can see a big peak or spike here and a second one here. This is unique to the cancer. It's not found in her healthy DNA. First blood sample, second blood sample, completely clear. So again, in this patient, I can with confidence say that there's no evidence of cancer from the blood test. This patient is essentially cured. And these samples, the second sample here was taken 9 years after surgery and treatment. And this patient remains disease-free, and it's now 12 years since she had her primary surgery and treatment. So we're extremely excited by this approach, but this is an approach in the research setting, and the strategy which we use cannot be rolled out into the NHS in its current format. So we need to make simpler, more focused analyses such that we can do this. And you may or may not have been aware of some publicity surrounding this last October, which was basically launched by Cancer Research UK, because we've commenced a study to roll this out into the breast screening programme. Now, the breast screening programme's available to all women over 50, where you request to come in and have a mammogram, see if there's any evidence of cancer there. Mammographic screening is good, but it's imperfect, and we want to know whether our blood tests can improve on that. Can we pick up cancer more specifically? Can we pick it up earlier? And if we pick it up, can we monitor whilst patients are undergoing treatment? And then with Julian locally here and funded by Hope Against Cancer, we've been working with these high-risk breast cancer families to see whether our blood tests are similarly useful there. Can we identify in someone who carries a BRCA1 mutation whether there's any evidence of cancer in your body or not? And similarly, other high-risk cancer families who may not have a BRCA1 or BRCA2 mutation, can we identify evidence of breast cancer by our blood tests? And this got a lot of attention, all the national papers and the TV were here, the radio were here. We were bombarded for a few days, but the study's now ongoing, and apologies if this doesn't work, but I got one of my students to upload the video from YouTube, which tells us about the study. Ooh, it's very quiet, isn't it? Okay, so this is Professor Charles Coombs at Imperial College, who is the lead clinician on our breast screening study. And through a single hospital, I will stop that because you're not going to be able to hear it, through a single hospital site, Charing Cross Hospital, we're recruiting women who are attending for their routine breast screening, asking them if they're prepared to give us a blood sample, take a single blood sample, and then over a 5-year period, we'll look at hundreds of different blood samples and see if we can improve on breast screening by our blood tests. Similarly, we're setting up another study. Once women have been diagnosed with breast cancer, we'd like to follow them over a 5-year period with blood tests, again see if these can help with monitoring outcomes and response to treatment. So I'll just quickly scroll through to the end. Oops, it doesn't like it, does it? It's not going to allow me to go on, sorry. So that's where we are. The future's very exciting, but all this takes a lot of... sorry, it's not going to allow me to go beyond that PowerPoint slide, I don't think. I'll just leave it up like that. A lot of this takes a lot of time and a lot of cash to resolve all these questions. All these studies are long-term. They take several years to come to fruition. We need, at the moment, to expand numbers. We've got good data from 65 breast cancer patients. We need to look in hundreds to know if all our tests will be successful in the future. So that's why we need this core, significant, substantive funding to allow us to do this. And Cancer Research UK funded my lab for the next 5 years. This is the group that's currently funded by Cancer Research UK. In my lab, I also have 2 staff at Imperial College in London. We've also got Hope Against Cancer funded staff as well. So finally, just to thank you, to all cancer families and friends for your support and for your fundraising, because without that, none of this would be successful. Okay, thank you. Any questions?
What are obviously the blood tests they're doing at Charing Cross?
Charing Cross,
yeah. Is that the same analysis of what you've been doing with your research?
That's right. It's a research-based study at the moment, so nothing's going to change in terms of a woman's, treatment and outcomes. It's just when you come for breast screening, women will be asked, are you prepared to give a single blood sample for our research study? Then if that woman is shown to have cancer, we'll be able to correlate our blood markers with the presence of cancer and the type and stage of disease. And then within that, there will obviously be a lot of women who don't have any evidence of cancer. So they're expanding our control population. So within the healthy population with no evidence by mammography, no evidence on follow-up with cancer, what do our blood markers look like? So it's those big studies which will give us the data that will allow us to then say, right, in 5 years' time, let's replace mammography potentially with an annual blood test. That's where we're going.
Will you be obliged to inform women if you find that they have cancer in their blood tests?
That's an interesting question. Finding whether someone's got cancer will be done in the normal way by the follow-up because of suspicious mammogram, taking a tissue biopsy, routine pathology diagnosis. We won't be changing that pathway at all. It'll just be correlating our blood markers with those outcomes. So no, we're not going to change or inform. And when women sign to give their consent, one of the boxes says that this doesn't change my pathway.
Is it like the biomarkers that we've already done, that have already been done?
Which
The
biomarker?
test that was already done.
For the study,
Yeah.
this is the
Yeah,
study,
so
yeah.
in the Julian Barwell study,
Yeah.
so the local Leicester study, obviously you and your families have consented to give a blood sample, so we're looking at your blood samples as well. So are the profiles similar or different in BRCA1, BRCA2 and other high-risk cancer families? And if we see them, when do we see them? Do we see them 5 years before cancer, 10 years before cancer, or only once you've got physical evidence of cancer in your body? So that's what we're doing there.
Yeah, we hope in the future that these sorts of tests will be useful alongside standard screening, alongside the genetic counselling, looking at family histories, looking at the genes that you've inherited. So that could become particularly important if we're starting to move into the preventative treatment phase, be it through chemoprevention from the work that Lynn's doing, or through things like tamoxifen. So can we see changes that are occurring in the body?
I think you answered what I was going to ask, but how far are you away from the screening phase?
What you mean, rolling it out into breast screening?
That's right, yeah.
So that my challenge is to be at the end of the programme, which is 5 years, to say we've got enough evidence or not to suggest that this should be rolled out into the breast screening programme. So it's a 5-year study basically. And it may be that in 5 years we'll be 50% of the way there, but it may be that we'll be 100% of
Just
the way
have to convince
there. Yeah.
the politicians that
Exactly.
it's a feasible thing to do.
But I think as Joyce showed with this 1,000 genomes project, the technologies, the advances that we've had in the last 5 to 10 years are so phenomenal that we can't really stare into a crystal ball, and I suspect that this approach will be implemented in the NHS within the next 5 to 10 years, not just for breast cancer but for all cancers. I've only focused on breast cancer. Everything I've said about breast cancer, you can do the same in lung cancer, pancreatic cancer, melanoma, bowel cancer. So this blood screening and monitoring approach is likely to be rolled out in terms of cancer diagnostics in the future.
So what you're talking about, how does that compare monitoring somebody's CA125
Right.
levels in ovarian
That's
cancer?
a very good question. CA125 has its benefits and it's used routinely in the States, it's used routinely in Europe, it's only used in ovarian routinely here. We're hoping our markers are better, more specific, but with a study I'm setting up now between Leicester, Manchester and London will do exactly that. Post-surgery and treatment, 6 monthly blood monitoring. How do our markers compare to CA125? Again, that'll be a 5-year study that we'll have to wait for the outcome.
What we say generally in clinic about markers like PSA and CA125 is that they are generally better for people who have been diagnosed already to give a feel for whether or not the treatment is working or if the tumour is coming back. They're not always so good at telling you up front if someone's actually developing a tumour. So it's really the rate of change of those markers which is more relevant rather than an individual reading. So, and in my experience, when we worked at the Marsden, we saw 4 women with ovarian cancer, and at the beginning they all had normal CA125s. As Jacqui says, the ability to use CA125 alone to pick up early ovarian cancer, the evidence is fairly limited, but we have one of the gynecologists here who may want to say more, so I won't say anything further at the moment.
I think the other point is that these single markers don't tell you enough information, basically. They're like a low-grade screen, but the idea with molecular profiling is you get a lot more information in a single look, which tells you a lot more detail about how the cancer's behaving at that time.
The final point about young women who are undergoing breast screening is that we know mammograms alone are not very good at picking up all tumours in young women because the breast density is much higher. And so I think in familial disease, there's even more reason to try and develop a blood test, because mammograms alone are not going to be the answer.
Can you just how often would these blood tests be taken?
Well, at the moment, the studies we're doing in breast screening, we're just taking a single sample. So women have 3 yearly recall, don't they? But we wouldn't then monitor those patients in the future. But the idea is, if it's successful, as with breast screening, we could align a blood test rather than a mammogram. So it might be every 3 years you have a blood test. If you're high risk cancer family, you might have an annual blood test from the age of 20, 25. That's the sort of approach we would want to develop.
We've got the ethics to look regularly at
Yeah.
BRCA carriers. It's just a matter of sorting out further funding, but it's something that could be considered.
Yeah, I mean, the vision is to get cancer like diabetes. It's, you know, it's lifetime manageable because we pick it up early enough and we monitor it, so then we can treat it better. Hi.
Hi. Are you doing any comparisons with what's found through the breast screening programme or anything alongside the research? Or
You mean the report that is
Yeah.
that what you're alluding
Yeah.
to? Yeah, I mean, I work with, well, the other David Cameron, not the Prime Minister, but David Cameron is a breast surgeon in Edinburgh, and he was one of the main authors of that report.
Yeah.
So the findings of that are that breast screening is worthwhile because it does lead to more lives saved. But there's this potential that some women may have inadvertently been overtreated. So they've had a mastectomy when a very early lesion may never have progressed to cancer anyway. So in our study that we're just starting at Charing Cross, all of those different types of alterations will be picked up in the samples as we go through the study. So we will be able to do correlations. And I've had discussions with David Cameron about this, and meeting him on Tuesday actually to talk about how we'll manage the data and what we're planning. So yeah. Okay. Well, thank you very much.